So most of the research in my lab centers around polycystic kidney disease, and that's a very common disease, a common genetic disease that affects about over 600,000 patients in the US, and it leads to kidney failure, typically around age 50 or so, which means that patients require either lifelong dialysis from that point on or a kidney transplantation. And there's currently no treatment available to slow down disease progression. And our research is aimed at trying to understand the mechanisms that lead to the disease. So it is known, the genes are known that are affected in the disease, but it's not actually quite known or well understood what these genes do, what the gene products do. So that's something we're trying to understand better. What are these proteins for? What is their purpose? What are the different mechanisms of their function? And why does disruption of these genes lead to the growth of thousands of cysts in the kidneys and destroys the kidneys? And we're trying to then come up with treatments to inhibit these pathways with drugs, so that we can attempt therapy, and we do this first in mouse models of polycystic kidney disease. We have these mice that form a type of the same disease that we see in patients, and we can test these therapeutic approaches in mice first. Yeah, so this was a really interesting way of how a collaboration can get started. We published a paper in PNAS back in 2006 on a discovery we had made in the lab, which turned out to be really important for understanding the disease, and it had to do with a signaling pathway called the mTOR pathway, which we found to be activated in polycystic kidney disease. And as it turned out, there was already a drug available that can inhibit this particular pathway. And this drug had been clinically used as an immunosuppressant for transplant patients. And it's a very specific drug, so it can very specifically inhibit this one kinase called mTOR. And we tested this drug in our mouse models of polycystic kidney disease and found that it was indeed very effective in reducing cyst growth in these mice. And in fact, it was probably the most effective drug that had ever been tested until then in polycystic mouse models. So we published this work, which suggested that this drug called rapamycin would be a very good candidate to test in clinical trials. And indeed it was tested also shortly thereafter. But once we published our data, it turns out that the head of research at a biopharmaceutical company called Endocyte, he read this paper and he had a particular interest in polycystic kidney disease because he has PKD himself. He's affected by the disease himself, and he saw our paper and saw, very long term what he thought would be a really good contribution he could make and his company could make to this research, which is to not just use this drug by itself, which is already known to have many effects that we don't want to have in patients, but to use the technology of his company to potentially target this particular drug to the kidney, which is where we want it to work. We don't want it to work in any other tissues in the body. And it just so turns out that his company has this technology where they can chemically link drugs to another compound called folate to make a conjugated drug, which then gets taken up in the body by what's called receptor-mediated endocytosis. And they had already known that this receptor is highly expressed in kidneys, and he thought, potentially this may be a way to target this drug to the kidneys. So he contacted me and we started some brainstorming on what we could do there together. And it was very exciting because I'd never been contacted by a head of research who has the disease that we're working on, of a company who has a technology that could actually help improve this drug. So I said, let's do it. He said, let's do it, and he came. He flew out here to visit us, and we refined our plan a little bit more, and then started this collaboration. And it turned out really. We got some really interesting results out of this that suggest that this new drug would be far superior to the unconjugated rapamycin. So the problem that came out in the rapamycin clinical trials is that essentially there was no beneficial effect on patients, and there were 2 large trials that were conducted in Europe with several hundred patients altogether. The limitation in these trials was that patients could be only given relatively low doses of these mTOR inhibitors, which is dictated by the known side effects of these drugs, which are very potent immunosuppressive drugs. And they also have other effects on wound healing, and there are side effects on skin rashes, for example. Which means that if you just keep increasing the dose of the drugs, patients would just drop out of these studies because the side effects would be too severe. It turns out that at the normal tolerable low doses of this drug, there's very likely not enough drug concentration that makes it into the kidney where it needs to work to be able to treat polycystic kidney disease. So this seemed to be the end of the road for mTOR inhibitors as therapeutic treatments in polycystic kidney disease, because it seems it's not feasible to just increase the drug concentrations to what we had been able to use in mice, which are much more tolerant to this drug. So the way around this now, probably the only way around this, is to be able to target these drugs directly to the kidney, which is exactly the approach we have been taking in collaboration with Endocyte. These compound drugs, or the compound version of rapamycin that we have tested with Endocyte, is able to be targeted to the kidney. Once inside the kidney, this drug is broken down again into the original drug, into the original rapamycin, which then has the effect locally on the kidney and not systemically on the entire body. So the next step is to we're drawing up a plan right now to verify the results that we have gotten using one particular mouse model of polycystic kidney disease. We would like to repeat it using a separate independent mouse model. And at the same time, we would also like to test several other similar compounds that Endocyte has already developed, which they will send us for this, so that we have a way of comparing slightly different versions of similar therapeutic compounds to see which one is superior. And depending on the outcome of the next study, hopefully one of these drugs will tell us this is the way to go. This is the drug that works the best in mouse models. And then it would be time to think about phase 1 clinical trials to establish the safety in patients first. And once that's established, it would go on to a phase 2 trial to test the efficacy of this compound in polycystic kidney patients.